Hello, my name is Christiane Herr. I am presenting this paper also on behalf of my co-author Yawen Duan. In this presentation, I introduce a first exploratory study on the ability of architectural facades to host biodiverse microbial communities. Our aim with this project is to inform the design of architectural facades that can positively contribute to healthy urban environments. In recent years, an increasing number of studies points to the importance of microbes in human health and well-being. Human beings are holobionts, hosting a great diversity of microbes in our bodies, among others in the gut, lungs, and skin. Even in the human body, human cells are outnumbered by non-human cells by a factor of 1.3. We don't know much yet about the interactions between these microbes and human health, but we do know they are closely linked, influencing conditions such as asthma, Crohn's disease, and the development of organs, such as the human brain. We also know that we need to maintain and support biodiversity in the microbiomes of our bodies and our environments for our future health and well-being. Most research on microbes in the built environment has aimed to kill and reduce their numbers. Most contemporary materials used in architectural facades are engineered to reduce the chance of microbes growing on them. This has significantly reduced microbial biodiversity on architectural facades. This project follows the calls of previous studies in biology and public health, suggesting a rewilding of urban environments to reintroduce microbial diversity. Not much is known about the microbiomes of our external built environments. In this exploratory study, we examine microbial growth on architectural facades. As a first step, we analyzed existing facades of a building complex on the campus of Xi'an Jiaotong Liverpool University in Suzhou, China. Suzhou has a mild and humid climate. The annual average temperature is 15.6 degrees Celsius, and the annual precipitation is 966 millimeters. The buildings were all built 10 years ago and include a variety of facade materials. We selected 15 sampling sites, including diverse orientations and weather exposures within the same climatic context. We took 2 sets of samples, one in December 2019 and one in August 2020. The building complex features not only typical architectural facade materials, such as glass and sheet metal panels, but also stone walls with water features, as well as walls initially designed as green walls. The green wall features did not develop as originally intended in the architectural design, as you can see in the images, but it resulted in building surfaces of moss-covered earth that contribute significantly to microbial biodiversity. We also took a control sample from the ground immediately next to the buildings. This diagram illustrates the relative locations of the samples taken for the study in December and August. You can refer back to this slide to identify the sampling site codes in the following diagrams. We outsourced the DNA analysis of the samples, conducting 2 analyses for each sample, including 16 sRNA bacteria and 18 sRNA eukaryotes analysis. Several samples were initially rejected, as they did not contain sufficient microbial material to conduct a full analysis. These include glass windows, metal facade panels, metal horizontal sunscreens, and artificial lawn. This in itself is a first result, as it indicates that these materials have very little capacity to host microbial life. Unsurprisingly, as most facade materials are engineered to repel microbes of all kinds for as long as possible, thus minimizing long-term maintenance requirements and surface changes. All remaining samples included some type of natural material, including timber, soil, and a variety of small plants. The DNA analysis generates results in terms of OTU, Operational Taxonomic Unit, where DNA sequences are clustered. We are interested in the diversity of taxa in the samples, as shown in the comparison in the image. 16 sRNA analysis shows bacterial diversity across the samples, and 18 sRNA shows eukaryotic diversity. The most common taxa have been named. For this brief presentation, we can only present the overall results of the analysis. We can state that overall diversity is larger in bacterial taxa compared to eukaryotic taxa across all the samples. There is a marked difference in microbial growth between summer and winter, with summer growth showing more common taxa and much reduced variety compared to winter. We assume this is related to heat stress on exposed surfaces. We can see that soil-containing samples feature more diverse microbial communities, with the control sample taken from the ground containing still more microbial diversity. We were surprised to find the greatest diversity on a timber enclosure of a planter, with the sample code 301 TPE. We are not sure yet why this is the case, but we suspect the presence of plants and insects could play a role. Generally, east-exposed surfaces feature more microbial diversity compared to west-exposed facades. It is tempting to ask, how can we support only desirable microbial taxa and selectively kill off the undesirable ones? We do not support this approach, as some taxa may be closely related yet have very different impacts on human health, as we explain in the paper. Instead, we argue for enhancing biodiversity more generally, to create artificial environments that are closer to the natural environments we evolved in and for. We can do this through architectural design. Surfaces can be designed to support diverse microbial communities all year round, based on the control of several environmental key factors. These, we call them design factors, based on results of the study, include the facade material, sunlight exposure, and the presence of soil. Results of the study also indicate that the presence of plants and insects plays a key role in enhancing microbial biodiversity, but we do not have sufficient data on these factors yet. This presentation can only give a brief insight into the rich findings generated by the study. Please do refer to the full paper for more details. The project will be developed further at the next stage. Based on the results, we aim to test diversity in microbial communities on purposefully designed and fabricated prototype panels. In the longer term, we hope that outcomes of the study can contribute to a new kind of facade design approach, complementing ongoing research initiatives that currently mainly originate from the sciences, in particular biology, environmental science, and public health. Thank you, and please do get in contact for any questions and comments you may have.